I'd like to thank Praveen and Maureen for the opportunity to present today. It's been 5 years since I've stood up on this podium, so I'm excited to tell you about what I've been doing over that time. So our technology was licensed from the Wilmer Eye Institute. It's a nanomedicine technology. It's been used now in 6 different clinical trials with no systemic safety events at all, which to me is amazing as a clinical developer. Over the last 30 years, I've never experienced this. What's unique about it is it only gets out of the vasculature where there's regions of inflammation, and it only targets those areas where there's inflammation present. We've been able to show preclinically in monkeys, rats, and mice that it does cross the blood-retinal barrier when there's inflammation present. We think we have some evidence that I'll share with you today that it does that in humans as well. And so we can now give drugs potentially systemically to treat eye disease without safety issues. And I'll have further information about that as I present today. We were able to show that it does get taken up by activated microglia and macrophage and RPE cells in human tissues as well, which is shown in the bottom image. And I don't have time today to show you the video. I really encourage you to go on our website, see the video. Everything in that video is based off human and animal data, and it does do exactly what's in that video, which is very unusual. I know you see a lot of videos, but this is a very unique thing. So it's called a hydroxyl dendrimer. It's on the far right bottom corner there. It's about 15 to 16,000 molecular weight and 3 nanometers in diameter. It doesn't bind to any serum or plasma proteins, doesn't get into any cells unless there's inflammation present. It's very, very unique technology. It's different than a lot of the things you might hear today in presentations because it is actually a drug. So we're not a drug delivery company. Just very, very important for you guys to recognize we're not a drug delivery company. This is a drug. It's not a prodrug. The whole thing is actually a drug, and I'll talk a little bit more about that. It does cross the blood-brain barrier as well. We're actually doing neurology studies in patients with neurological disease and hoping to show that it crosses the blood-brain barrier. It does not have any off-target toxicity because it doesn't escape the plasma compartment unless there's inflammation present, and we've not seen toxicity, as I mentioned, in humans and in chronic animal dog tox that you would normally expect for some of the compounds that we're using. In addition, it's very tunable, so we can do different approaches for oncology as well as some of the other indications. So this is the molecule itself. The warhead on the end, you probably recognize it's an analog of sunitinib. It's covalently linked directly to this hydroxyl dendrimer. As someone said to me at dinner last night, this is not your grandfather's RTKI because it is 500 times larger in size. It doesn't distribute the same way, and it still has all its original binding activity. So it can still bind to all the VEGF tyrosine kinases and PDGF and c-KIT and CSF1R with the same nanomolar affinity as the parent molecule. It is not metabolized or cleaved off. The whole thing is active. So again, it's not a prodrug. It's not a different approach other than that. Once it's taken up by the cells, we've been able to show, and we've presented this at previous ARVO meetings, it stays within those cells for a very long period of time and has an ability to shut down VEGF expression. And why is this important? Well, we think that if you can shut off VEGF expression, you can stop fluid from coming back into the eye. And so if we could have a long-term therapy where we stop fluid from coming back to the eye, this could be a huge advantage. And we've been able to show this not only subcutaneously, which we're testing now in the clinic, but we can also do it orally. So the whole idea is to actually replace the maintenance paradigm, which all of you I'm sure are very familiar with. After you go through the loading doses, you get rid of all that VEGF that's in the back of your eye. Then you want to make sure that the VEGF doesn't come back. Well, the way to do that is with our drug. By giving it systemically, either with a sub-Q or oral dosage form, we think we can prevent VEGF from coming back into the eye and needing to have to rescue patients with an anti-VEGF therapy. So the whole paradigm that we're hoping for is an at-home administered product. Of course, this would be an advantage for the patients and to some physicians as well, where they'd be able to give this drug at home. And ultimately with an auto-injector, so patients with arthritis and diabetes already take this type of injection, so they think they'd be fairly familiar with it. Obviously, oral will be better, and we hope to be in an oral dosage form sometime next year. And the other thing that's an advantage with systemic administration is we target both eyes. So there have been studies with RTKIs where they've been able to show they get both eyes treated from systemic administration. We hope to see the same thing. This is a huge market opportunity, as you know, maintenance therapy, and we've already done quite a bit of market research with physicians and patients to support that. So the first study that we completed in patients, we did finish a healthy volunteer study. Again, there was no systemic toxicity, and in chronic animal studies through 6 and 9 months, there were no liver toxicities. And what we're able to do here with the help of Peter Campachero, we designed a very different study from what you might see the rest of today. We took patients that have already responded to anti-VEGF therapy. In our trial, we gave them an intravitreal injection of aflibercept. Over time, we looked at when the fluid came back to the retina, and then we dosed sub-Q with our drug. So each patient becomes their own control. We know exactly how long it takes for fluid to come back into their eye. And so what we're able to show in this study, we did about 12 patients, 7 wet AMD and 5 DME patients. It was safe and well tolerated. We didn't see any liver enzyme elevations, anything that would suggest there's any toxicity systemically. And what we did see is in 11 out of those 12 patients, for 2 weeks, there was no fluid change in the eye and no BCVA change. For 9 out of those 12 patients, for 4 weeks, there was no fluid change or BCVA change. In 4 patients out of the 12, there was 12 weeks, so that had been 24 weeks since they got an aflibercept injection. There was no change in fluid or BCVA. So I think that's pretty compelling initial evidence that says we're probably getting the drug to the target. Now can we sustain that effect? So the study that we just recently announced that we're dosing in is the one shown here. Again, these are patients that have already responded to anti-VEGF therapy, wet AMD or DME. They go into our study. We use them as their own control by giving them intravitreal aflibercept. Once fluid comes back into their eye, we're going to give them a dose of aflibercept to get rid of that fluid, and then give a chronic dose of our drug to prevent any fluid from being generated in the back of their eye. And so we hope to be able to share this safety and efficacy data this summer at the Retina Society meeting and potentially at AAO, and look forward to speaking with everyone during the networking break. And thanks again for the opportunity to present today.